A warm welcome to all of you. Today we are discussing transpiration in plants. Transpiration is a very useful process for plants. What are the uses of transpiration, or what are the significance of transpiration? First point, creating suction force in the stem to enable the roots to absorb water and mineral nutrients, creating suction force. This point we already discussed in the last video, right? Then the second significance, that is for cooling. Then two main significance are there for transpiration, creating suction force in the stem. Then the next point for cooling. These are the two main uses or significance of transpiration. All plants continuously absorb water through their roots. Here you can see. This water is conducted upward through the stem and is distributed to all aerial parts including the leaves. Right. Then what is the definition of transpiration? Transpiration is the process of loss of water in the form of water vapor. Very important. Loss of water in the form of water vapor from the leaves and other aerial parts of the plant. Then two main points you have to remember here. This is the process of loss of water in the form of water vapor. Water vapor from where? From the leaves and other aerial parts of the plant. Demonstration of transpiration. For demonstration of transpiration, we are discussing two experiments. Experiment 1. Take a medium-sized, well-watered potted plant. Cover the plant with a transparent polythene bag and tie its mouth around the base of the stem. Leave the plant in sunlight for 2 to 3 hours. What will be the result? You can see drops of water will soon appear on the inner side of the bag. What is the reason for this? Water droplets. Here, water vapor condensed to form water droplets or water. What is the definition of transpiration? Transpiration is the process of loss of water in the form of water vapor from the leaves and other aerial parts of the plant. Then this here water vapor condensed to form water droplets. This is the experimental setup. And what will be the control setup? For control setup, a similar polythene bag with its mouth tied and kept in sunlight will show no drops of water. That is the control setup. Then how do you test these drops of water? If tested with dry cobalt chloride paper, you have to know that this dry cobalt chloride paper is blue in color. Then you can say dry cobalt chloride paper or blue cobalt chloride paper turns pink when it contact with water. Then these drops of water, then what will be the result? Dry cobalt chloride paper turn into pink. In this way you can confirm these drops are water. Another experiment for the demonstration of transpiration is bell-jar experiment. In bell-jar experiment, we have to arrange three setups: setup A, setup B and setup C. In setup A, take a small well-watered potted plant and enclose the pot completely within a polythene bag. Here you can see that. and tie the mouth of the bag firmly around the base of the stem. Then, why we are doing this? To prevent the loss of water vapor from the pot. Very important point, because this is a well-watered plant. There is a chance to escape water vapor from the pot to outside. So to prevent that, we have to cover the pot with a polythene bag. Then what you have to do? Cover the entire plant under a bell jar. In setup B, arrange another plant, another similar plant, and cover it with a bell jar. But we have to place an indicator of moisture. This is the indicator of moisture is cobalt chloride paper. Cobalt chloride paper is an indicator of moisture. We have to place a dry cobalt chloride paper. Here is dry cobalt chloride paper is blue in color. Then a blue cobalt chloride paper we have to place inside the bell jar in setup B. Then in setup C is the third bell jar without plant. We don't keep any plant inside the bell jar in setup C, but we have to place a dry cobalt chloride paper, or that is blue in color, right? Yes, inside the bell jar in setup C. Then after half an hour or after 1 hour, what will be the result? The first bell jar, here you can see, is water vapor condensing on its inner walls. You can see that, right? Then in setup B, you can see the water droplets or condensation of water vapor. You can see is then this dry cobalt chloride paper turn into pink. You know that the last slide we already discussed. Cobalt chloride paper pink when exposed to moisture. Then this blue cobalt chloride paper turn into pink in setup B. Then in setup C, what will be the result? There is no plant, that's why there is no moisture. Then this blue cobalt chloride paper will not change into pink because of the absence of moisture or absence of water droplets, because there is no plant. Then here you can see two visual proof of transpiration. First one, condensation of water vapor into droplets, water droplets. Here you can see that in setup A and setup B. There is no water vapor in setup C. Then here another proof, which is another proof, change of color in cobalt chloride paper. In setup B, you can see pink cobalt chloride paper because of the presence of this moisture, water droplets, is blue cobalt chloride paper turn into pink. Then in these three setups, which is the control setup? Can you say that is third bell jar is the control setup, because you know that there was no moisture in the air due to transpiration because there is no plant. The next topic is measurement of transpiration. For this we are discussing two different methods, weighing method and potometer method. In weighing method, we are using glass bottle linked by a rubber tube to a graduated side tube, then filled with water. Pour 2 to 3 drops of oil above the water. Why we are doing this? to prevent the loss of water by evaporation. This is very important. Then what you can see after 1 hour or after half an hour, what you can see, the water level in the side tube falls. Here you can see that. To demonstrate loss of water through transpiration from the leaves. This would indicate the volume of water loss. Here that can be compared with the loss in weight with the help of weighing machine. Here you have to weigh the experimental setup in the beginning, then after 1 hour or after half an hour you have to weigh again. Then we can see the difference, right? The loss of weight by the plant during that time is due to the loss of water by transpiration. Another weighing method. For this weighing experiment, we have to use one test tube filled with water and inserting a leafy shoot. Very important, without root. Then inserting a leafy shoot in it and pouring some oil on the surface of water to prevent the loss of water from the test tube to outside by evaporation. Very important point. You have to remember this for give reason question. Then you have to weigh the test tube using spring balance, or you can place the test tube in a small beaker and weigh them together. Then remove the test tube and keep it in straight in the test tube stand for few hours. Then wait again after few hours, after 1 hour or after 2 hours. You have to wait again by keeping in the same beaker, or you can use spring balance. Then you can see the difference. Any difference in weight will indicate loss of water by the shoot due to transpiration. Since there is no roots, you can see there is no roots here. Let's say there is no active absorption of water. Then the loss of water through transpiration will be very less, will be much less. Then here you have to know pouring some oil on the surface to prevent the loss of water by evaporation. That is very important point. Yes. Then another point you have to remember here, difference in weight will indicate the loss of water by the shoot during transpiration. Potometer is another important apparatus for the measurement of transpiration. Potometer is a device. What is a potometer? Potometer is a device that measures the rate of water intake by a plant. Then these potometers do not measure the water lost during transpiration, but measure the water uptake by the cut shoot. Then why we are using potometer? For the measurement of transpiration. Yes, because this water intake is almost equal to the water lost through transpiration, lost during transpiration. That's why we are using potometer for the measurement of transpiration. Here you can see GANONG'S potometer is a twig of some suitable plant. Any plant. As example, coleus, cut with a sharp knife, is fixed in an apparatus as shown in the figure. The entire apparatus is filled with water. We can fill water through reservoir, then so that no air spaces are present. An air bubble. An air bubble is introduced into the horizontal graduated capillary tube. Here you can see this is air bubble which is dipping into the beaker containing water. How we can introduce this air bubble by lifting the bent capillary tube above the colored water so that air may be sucked in due to suction pull and is again dipped into the water. As transpiration proceeds, what you can see, or as the water is lost from the twig, a suction force is set up which pulls the water from the beaker. The suction force and transpiration pull we already discussed in the last videos, right? Yes, a suction force is set up which pulls the water from the beaker, and the bubble in the capillary tube moves along. Then the reading on the capillary tube would give the volume of water lost in the given time, right? Then to repeat the experiment, the air bubble can be brought back to its original position. How we can is brought back the air bubble in original position? By releasing some more water from the reservoir. This is reservoir. Then reservoir into the capillary tube by opening this stopcock. Then you have to remember, photometers do not measure the water lost during transpiration, but measure the water uptake or water intake by the cut chute. Yes, that's why we can say some of the water is used by the cell to carry out other processes. That's why we can measure only water intake, not water lost through transpiration. We can't measure water lost during transpiration. Precautions in the use of potometer. What are the precautions for the use of potometer? First one, the potometer should be made completely watertight. Then another precaution, the twig should be cut obliquely. Here you can see we have to make an oblique cut to allow a larger surface for water intake and underwater to avoid suction of an air bubble into the twig, which will stop the absorption of water into the side. What are the limitations in the use of potometer? Yes, first one, introducing an air bubble is not very easy. The second point, the twig may not remain fully active for a long time because there is no root, only leafy shoot you can see here. That's why the twig may not remain fully active for a long time. Then another limitation, any change in the outside air temperature may affect the position of the air bubble in the capillary tube. Based on the temperature, is the position of the air bubble will change. Revision. Answer the following. First question, mention any two limitations in the use of potometers. First one, introducing the air bubble is not very easy. That is the first limitation. Second one, the twig may not remain fully active or fully alive for long time. These two limitations you can write here. Then the another question, transpiration is best defined as. Here they are giving three options. You can choose the best one. First option, loss of water from the plant. Here from the plant means roots also included. That's why we can't choose this. Second option, loss of water as vapor from the plants. Here also roots included here, that's why we can't choose. Then third option, evaporation of water from the surface of leaves. This is the best option. Is C we can choose as answer. Right. Then what is the definition of transpiration? Transpiration is the process of loss of water in the form of water vapor. Here they are giving that from the leaves. Here they are giving on the surface of leaves and other aerial parts of the plant. Here they are giving, then this one is the best answer for this question. Thank you for watching. If you like my channel, please like, share and subscribe. Thank you.